Thank you so much for this brief introduction, and I thank the organizers for inviting me to present some of our research results before you. So today we'll be talking about the role of a specific microRNA, the microRNA 9 and the MCPIP1 axis in the regulation, post-transcriptional regulation of IL-6 expression in OA chondrocytes. So you have heard it many times, and it's mandatory for me to go over this again, that OA is the most common form of joint disorder and the dominant one among the elderly. The disease mostly affects the large weight-bearing joints and is characterized by the occurrence of chronic pain and limitation in the range of movement. And the hallmark of OA is the focal degradation of the cartilage, which is also associated with low-grade inflammation. The monocyte chemoattractant protein 1 induced protein 1, or MCPIP1, is a multifunctional protein that actively participates in several distinct signaling pathways. It downregulates LPS-induced inflammatory response by acting as an RNAs. It inhibits JNK and NF-kappa B signaling. And recently it has been shown that MCPIP1 deficient mice display fatal disseminated inflammation. And MCPIP1 deficiency also causes abnormally high levels of IL-6 in the serum. So what is the relationship between OA and IL-6? The higher BMI and elevated serum levels of IL-6 correlates with the development of radiographic knee OA. IL-6 is a pro-inflammatory cytokine that activates the transcription of its target genes via formation of an IL-6 receptor complex. IL-6 is produced by chondrocytes and synoviocytes upon stimulation with IL-1 beta. OA synovial fluid contains high levels of IL-6, and IL-6 induces the expression of MMP13, and it inhibits type 2 collagen synthesis. So our hypotheses were high levels of IL-6 in the synovial fluid of arthritic joints suggest dysregulation of IL-6 expression in OA. The second hypothesis was, as deficiency of MCPIP1 has been linked to abnormal production of IL-6, we further hypothesized that MCPIP1 expression and function is inhibited in OA. So we did the immunohistochemical analysis and the gene expression analysis, MCPIP1 mRNA expression. In the undamaged cartilage, it was high. In the damaged cartilage, MCPIP1 expression was low. And IL-6 expressing chondrocytes were abundant in damaged cartilage, but not in the undamaged cartilage from the same joint. So, and when we stimulate the human chondrocytes, which were isolated from the OA cartilage, obtained from the patients who underwent total knee arthroplasty, as long as the MCPIP1 expression was there, the expression of IL-6 was not high. But when the MCPIP1 expression went down, the IL-6 expression took off. And at the mRNA level, as well as this was evident at the protein level, and the secretion of IL-6 in the culture medium as determined by an IL-6 specific ELISA. And this is an in-situ immunofluorescence hybridization using the RNAScope technology, again showing the negative correlation between the expression of MCPIP1 and IL-6. So when MCPIP1 is high, IL-6 is low, but when MCPIP1 is low, the expression of IL-6 is high. Again indicating that this protein is somehow involved in the regulation, in the post-transcriptional regulation of IL-6 expression. So when we knock down MCPIP or overexpress it in human chondrocytes, the expression of when we knock down, there is very little expression of MCPIP1, and when MCPIP1 is low, there is tremendous amount of the IL-6 production. MMP13 production also goes up, but aggrecan and MMP3 and Col10A1, their expression is not affected by MCPIP1 knockdown, indicating that MCPIP1 is not involved in the post-transcriptional regulation of these genes. And when MCPIP1 is overexpressed, the expression of IL-6 goes down, MMP13's expression is going down, and again MMP3, Col10A1 and aggrecan, these genes are not affected by overexpression of MCPIP1. And when we checked for the stability of IL-6 mRNA in chondrocytes with knockdown or overexpression of MCPIP1, the stability, when MCPIP1 was knocked down, the stability of IL-6 mRNA was significantly higher than the control chondrocytes. And when MCPIP1 was overexpressed, the stability of IL-6 mRNA went down, again confirming that this protein is involved in the post-transcriptional regulation of IL-6 expression. Then to prove that MCPIP1 binds IL-6 mRNA in human chondrocytes, we did the RNA immunoprecipitation, and as you can see, when MCPIP1 bound to the IL-6 mRNA, when this was immunoprecipitated, it brought more of the IL-6 mRNA as compared to the control IgG. And MCPIP1 also binds to its own mRNA and regulate its expression, so there was some MCPIP1 mRNA as well present in the immunoprecipitates. The expression of both MCPIP1 and IL-6 is regulated by NF-kappa-B, as shown here by when we use this NF-kappa-B inhibitor, the expression of both IL-6 as well as MCPIP1, it goes down. So now the post-transcriptional regulation of MCPIP1 expression, if you remember from one of the previous slides that after about 8 to 16 hours of stimulation, the MCPIP1 started going down, showing that this gene is also post-transcriptionally regulated because its stability of its mRNA was not affected. So we looked at microRNAs, which are small non-coding RNAs that suppress gene expression by inhibiting translation, promoting mRNA decay, or both. The mature microRNA guides the microRNA-induced silencing complex to partially complementary sequences or microRNA recognition elements in target mRNAs to repress expression. And when we did the bioinformatic analysis, we identified the MIR9 seed sequence or MRE present in the 3' untranslated region of MCPIP1 mRNA. So IL-1 beta is a potent inducer of MIR9 in human chondrocytes, and its expression was also higher in the damaged cartilage as compared to the smooth cartilage. And the expression of MCPIP1 was low in damaged cartilage compared to the smooth cartilage, if you remember from one of the previous slides. Expression of MIR9 is also regulated by NF-kappa-B in IL-1 beta stimulated human chondrocytes. It's not regulated by other signaling pathways like P38 or ERK or JNK. So when we overexpress MIR9 mimic or the MIR9 or the microRNA or its inhibitor, the MCPIP1 and IL-6 expression is altered. So when MIR9 inhibitor was used, which inhibited the function of microRNA9, the expression of MCPIP1 went up. When we used the MIR9 inhibitor, the expression of the IL-6 mRNA went down. And this is also evident from the IL-6 production. So again, it shows that the MIR9 is indirectly regulating the expression of IL-6 because it has no binding site in the IL-6 mRNA. the binding of the microRNA with its seed sequence in the 3' untranslated region of MCPIP1 mRNA was verified using a luciferase reporter construct. So when the construct and the miR9 were co-transfected in chondrocytes, the activity of luciferase went down, the reporter gene went down. But when the binding site or the seed sequence was mutated, there was no effect of miR9 when these were both, the mutant and the microRNA9 mimic were co-transfected in human chondrocytes. And very interestingly, IL-6 also induces the expression of miR9 in OA chondrocytes. And initially, the IL-6 is produced in low amounts when MCPIP1 is high. So it's a sort of SOS mechanism that IL-6 now inducing the inhibitor of MCPIP1 in human chondrocytes. So we conclude that expression of MCPIP1 and IL-6 is inversely correlated in smooth and damaged OA cartilage. And the expression of MCPIP1, IL-6 and miR9 is induced upon IL-1 beta treatment via activation of the NF-kappa B signaling pathway. And in addition, IL-6 also induces the expression of miR9. miR9 or siRNA mediated downregulation of MCPIP1 leads to increased expression of IL-6 and MMP13, and overexpression of MCPIP1 or miR9 inhibitor blocked IL-6 and MMP13 expression. Our findings thus suggest that miR9 mediated downregulation of MCPIP1 acts as a molecular switch for the enhanced expression of IL-6 in OA chondrocytes. And this work was supported by these grants from the National Institute of Health and funds from the Northeast Ohio Medical University. And this is our campus, and this is our research building. Thank you very much.